Hello everyone, welcome back to my channel. It's been a long time. So I am here with the session on the development of spinal cord. It's very important as far as UG or PG is concerned. So we know the spinal cord is developed from the caudal end of the neural tube, and neural tube is formed from the neuroectoderm by the after the formation of the notochord, which acts as a inducer of the neural tube. So we know the neural tube will be formed by the folding, and what happens after that? We can see that this is the neural tube, and in the beginning the walls of the tube are very thin, and it is enclosing a cavity known as neurocele. And later what happens is, by around 5th week of development, the epithelium or the walls of the neural tube will be redefined. That is, the lining of the neural tube is known as matrix. And what happens to the matrix? So you can see that this is the neurocele, the cavity, and around the neurocele, first we can see that the wall of the neural tube, which was very thin in the beginning, develops as the matrix or the matrix zone, otherwise known as ependymal zone. And what is happening in the matrix? Two types of cells are formed within the matrix. One is neuroblast and the other one is spongioblast. So neuroblast gives rise to the neurons, and the spongioblast will give rise to the neuroglial cells. So the neurons and their supporting cells, neuroglial cells. Now what happens to the neurons? The cell bodies of the neurons will actually migrate just periphery to the matrix, and they will be forming the mantle zone. So it is the first zone is the matrix zone. Then you have the mantle zone just outer to it, where the cell bodies of these migrating neurons are concentrated, and that will be forming the grey matter of the spinal cord. We know the spinal cord has got the grey matter inside and the white matter outside. Now what happens to the axons of these neurons? The axons of these neurons will occupy the outer zone of the mantle zone, that is known as marginal zone. So three zones are developed from the walls of the neural tube. The innermost, the matrix zone or the ependymal zone. The outer, just outer to it, we have the mantle zone, where we have the cell bodies of the migrating neurons, which forms the grey matter of the spinal cord. And outer to it, we have the marginal zone, which is mainly occupied by the axons of these neurons, which are concentrated in the mantle zone. So we have three zones for the developing spinal cord. Now one more thing which I would like to mention here is the remaining matrix after the formation of the neuroblast and spongioblast will be forming the ependymal lining of the central canal of spinal cord. That is the fate of the matrix zone. Now recent theories propose that there are no three zones like this, and according to that recent theory, which is put forth after a detailed study of the electron microscopy, they say that the neural tube is made up of pluripotent neuroepithelial cells. Pluripotent means the cells which can give rise to any types of cells, and it is just a pseudo-stratified neuroepithelium. And this layer is actually giving rise to neuroblast, and their neurons will be seen, the cell bodies of the neurons will be seen in the mantle zone, and the nerve fibers will be seen in the marginal zone. So ultimately, the previous theory, the classical theory, comes into existence, but the only difference is according to the new theory, there is no matrix zone, but they believe that it is just made up of a pluripotent neuroepithelial cell, and the different zones which we see are not actually zones, they are actually the interkinetic migration of these neuroblast cells. So that is just an additional point for the postgraduates. Now we can see that there is a dorsal root ganglia which is connected to the spinal nerve emerging from the spinal cord. And the central process, the process of the dorsal root ganglia which is going to the spinal cord, either ascends in the white matter, that is the marginal zone, or sometimes they synapse in the mantle zone of the concerned region. So that is what happens for the central process of the dorsal root ganglia. They either ascend in the marginal zone, which is actually the white matter of the spinal cord, or they synapse with the mantle zone. So this is just an overview of what happens to the walls of the neural tube. Simultaneously, we should also consider something about the functional column of the spinal cord. How this grey matter is redefined as the functional columns. When we think about the functional columns of the spinal cord, we have only general columns. So that means there are some special columns in the brain stem. That point you have to remember, there are 3 special columns in the brain stem, but when you think about the functional columns of the spinal cord, we have only general columns. Now when we take a cross section, we can see that the cavity is just like a slit, so that the roof and floor are very thin plate, and you call it as a roof plate and the floor plate. And what happens to the lateral wall? They are actually thickened. The roof and floor are very thin, whereas the lateral walls are thickened. And there is a sulcus which separates this lateral wall into a dorsal lamina and a ventral lamina. That is how the spinal cord gets its shape. Okay, so the sulcus limitans divides the lateral wall into a dorsal lamina, otherwise known as alar lamina, and the ventral lamina, otherwise known as basal lamina. Now we know the dorsal part of the spinal cord is actually concerned with the sensory, and the ventral part is concerned with the motor. So the functional columns are also like that. In the dorsal or alar lamina, you have the efferent columns, which are mainly sensory. And in the ventral part or basal lamina, we have the efferent columns or motor columns. That is one important thing which you should be knowing. Now we already mentioned that when you think about the spinal cord, the functional columns, we have only general columns. Now let's see what are the columns in the alar or dorsal lamina. There are 2 general columns. One is general somatic efferent. The other one is general visceral efferent. So general somatic efferent is the dorsal most, and next to that you have the general visceral efferent. Where will you get this general somatic efferent? This is seen throughout the spinal cord, from the beginning of the spinal cord till the termination of the spinal cord. But when you think about general visceral efferent, this column is confined to the thoracic region, lumbar region and sacral region. In the cervical region, this column is not there. So why you don't get this general visceral efferent column in the cervical region? Don't we have blood vessels and the viscera there? That is actually by the cranial nerves coming from the brain stem, and we have special columns there. Now coming to the basal lamina. Basal lamina is mainly efferent. E means motor, going to the effector organ. There also you have 2 columns, one going to the viscera and blood vessels, that is general visceral efferent, that will be lying closer to the general visceral efferent. And where will you get this general visceral efferent? That again will be seen only in the thoracic, lumbar and sacral regions. Whereas the ventralmost column is known as general somatic efferent, and that is seen throughout the spinal cord, and they supply the skeletal muscles. So what about the general somatic efferent? So once again I will just give a clear view. The dorsal or alar lamina, ventral or basal lamina, all the functional columns of the spinal cord are general. In the brainstem only you have special columns. Now the dorsal is mainly sensory, ventral is mainly motor. This much is clear, isn't it? Sensory. Sensory means the sensations. They are mainly superficial and deep. Superficial sensations are mainly cutaneous sensations, and deep sensations are proprioception. These are going through the general somatic efferent column, which is there throughout the spinal cord. Now general visceral efferent, that is carrying sensations from the viscera and blood vessels, and they are not seen throughout the spinal cord but only confined to the thoracic, lumbar and sacral region. Coming to the ventral or motor, that is efferent. So you have general visceral efferent, which is going to again the viscera and blood vessels, which is lying closer to the general visceral efferent, and it is not seen throughout the spinal cord, it is seen only in the thoracic, lumbar and sacral region. Now the ventralmost is general somatic efferent, that is actually motor, and it is seen throughout the spinal cord, and it is supplying the skeletal muscles. So in a nutshell, we know the spinal cord is developed from the caudal end of the neural tube. After the neural tube is closed, once we first and in the beginning it is very thin lining, then it is modified as zones, the 3 zones according to the classical theory, matrix zone, Mandel zone and marginal zone, and according to the new theory proposed, it is nothing but the epithelium, the pseudostratified neuroepithelium, which is mainly pluripotent, and the ones which we see as Mandel zone and marginal zone are nothing but the different different times of migration of the neuroblast cells which are formed in the pseudostratified neuroepithelium. And I will be doing one more session adjacent to this, which includes the positioning of the spinal cord and the common congenital anomalies. So keep watching, and at this moment I would like to mention about the ANASCULPT 2023. The details I will be putting in the section behind. Thank you.